One of the biggest problems is that we don't diagnose Alzheimer's disease until there are symptoms of cognitive impairment or early stages of dementia. And by that time, there's been a lot of damage to the brain. So the problem is we don't diagnose the disease until the wildfire is blazing. It's too late just to treat amyloid and blow out the match. That match was lit decades before. I think that this drug will be the statin of Alzheimer's. That in the future, based on your genetics, you will get a blood test. It will tell you if you need to begin this drug, just like you have a cholesterol test.
My voice in today's episode is powered by 11 Labs. Rudy Tanzi is a neuroscientist at Harvard who helped discover key genes linked to Alzheimer's disease and has spent decades trying to understand what actually drives brain decline. His work has reshaped how we think about memory, aging, and what might one day stop these diseases. And recently, Dr. Tanzi thinks they may have found the first real life-changing treatment that will forever change Alzheimer's. Dr. Tanzi, welcome to the show. Before we start with anything else, please tell us about this new treatment. How did it start? What does it mean for the disease? And how soon will it be in people at scale?
Well, thank you, Tim. It's a pleasure to be here and to share what we're doing in our battle to defeat Alzheimer's disease. And I should say that I think the way we're going to stop Alzheimer's is to detect it early on and intervene early, preferably pre-symptomatically. One of the biggest problems is that we don't clinically diagnose Alzheimer's disease until there are symptoms of cognitive impairment or early stages of dementia. And by that time, there's been a lot of damage to the brain that occurred over, on average, a 22-year span. So before someone goes to the neurologist and is diagnosed with Alzheimer's disease, for 20 or 30 years, that pathology was building up in the brain. Unfortunately, now we have approved blood tests that can tell us if someone is on their way. We have genetics that can tell you if you're at high risk. Many of the genes our lab discovered, we were involved in discovering the first Alzheimer's genes. Actually, it will be the 40th anniversary of finding the first Alzheimer's gene back when I was a student in August. And so we've learned from those genes what we need to do. But we've also learned that to nip this disease in the bud, to stop it, you have to start treating a decade or two or even three in advance. So the drug that you're referring to, Tim, is a drug I've been developing for 25 years. We spent about $70 million of investment from various companies that have come and gone, from the NIH, from foundations like the Cure Alzheimer's Fund, all combined. And just this past February, after this 25-year, tens of millions of dollars journey, the FDA gave us the authorization to start our first phase 1 clinical trial. The phase 1 trial would be to make sure this drug is safe. What does this drug do in a nutshell? It's aimed at safely lowering the production of the amyloid beta protein. Amyloid beta protein is a small peptide that's made in the brain that in excess, or if you have the more toxic forms of it being made, will create amyloid plaques. The amyloid plaques, I like to say, are like a match. They light the brush fires of what are called tau tangles that start spreading inside of nerve cells. That process over 20 or 30 years slowly initiates enough neuroinflammation. If the tau tangles are the brush fires, the neuroinflammation is like a wildfire. And then once you have that neuroinflammation wildfire going, that's when symptoms can begin. So the problem is we don't diagnose the disease until the wildfire is blazing. It's too late just to treat amyloid and blow out the match. That match was lit decades before. So what we need is a safe, affordable drug that people can take as soon as we know that match is lit, so we can put it back out before there's a wildfire. And that's what this drug does. It's called a gamma secretase modulator. Very different for those who are listening who know this field than a gamma secretase inhibitor, gamma secretase inhibitors came first. At Lilly, those were dangerous. They had side effects. The gamma secretase modulator is more nuanced. It's to avoid the safety issues. And we're very excited that I think that this drug will be the statin of Alzheimer's, that in the future, based on your genetics, you will get a blood test. It will tell you if you need to begin this drug, just like you have a cholesterol test. And it tells you whether you need to start a statin or a cholesterol drug to avoid heart disease. Just substitute amyloid for cholesterol, Alzheimer's for heart disease, our drug, gamma secretase modulator, for statin. That's how I think the future is going to be. So to answer your question, we're just starting the safety trial this year. And for disclosure, I had to start a company to do this at the request of the NIH. It's called Acta Pharmaceuticals. I'm the founding scientist. You don't want to put a new chemical entity into humans for the first time as an academic, so you should do it with a company or as part of a company. So that's what we're doing. The phase 1 would take about 6 months. And then when we move to phase 2, hopefully we'll do it with a pharma partner so that we can go 10 times faster. I started this company to stop Alzheimer's, not to make money. So I tell everyone involved, speed over greed. As soon as a pharma company is willing to work with us and take this process 10 times faster to get it to people sooner, we're on board with that.
The speed from them is just what, the resources they have?
Oh yeah, I mean, if pharma companies with, hundreds of billions of dollars of market cap and thousands of people are going to move a lot faster than a little startup biotech company. And so some biotech companies want to hang on and make more money. This is the exact opposite. As soon as we can unload this and let a pharma company take it and go 10 times faster, we're ready.
And you said like it doesn't have the same risks as the other one did. So is this something that healthy people could hypothetically take once in a while to make sure it never, and it sounds like it's far enough up in the chain that if the wildfire never starts, you miss Alzheimer's altogether, right?
Yeah. I mean, there's a controversy about amyloid and Alzheimer's that's still out there, because if you're diagnosed with the disease and you only hit the amyloid, it's like being in heart disease diagnosed with congestive heart failure and you need a coronary bypass. And imagine the doctor says, that's okay, we're just going to lower your cholesterol. You had to do that 20 years ago. It's exactly the same with Alzheimer's. You have to hit amyloid safely 10 to 20, 30 years before symptoms to avoid Alzheimer's later. And that's why there's controversy, because if you only hit amyloid in patients who are diagnosed clinically, that wildfire is going. You might slow the disease down because you're blowing out the matches that are going to start the next wildfires, but you're not going to make them cognitively better. And that's why there's some controversy. And it hurts us because every time journalists and the media write that amyloid isn't helping, they're not looking at the real question. It's not whether to treat it, it's when to treat it. And it's exactly similar to cholesterol and heart disease.
So if someone's taking this, whatever, 20 years before, because they have the blood, they get a blood test done, they're at risk for it, they start taking this, is it like what percent effectiveness is it? Is it 100%?
We predict that if, let's say you have a high genetic risk, like the early onset familial Alzheimer genes that my lab and other labs found 40 years ago, 30 years ago, those hard-hitting early onset familial Alzheimer genes like APP, presenilin 1, presenilin 2, they have mutations that guarantee the disease usually by 50 or 60. So if you have those mutations, you don't even wait for the blood test, for the biomarker. You just say, I need it now. If you carry 2 copies of the ApoE4 variant, apolipoprotein E4, also called ApoE epsilon 4 variant, it's in about 25-30% of the population. But it's a big problem if you have 2 copies. If mom and dad gave you an ApoE4, which is more like 5 to 10% of the population, then your risk goes up 14-fold with 2 copies of ApoE4. 1 copy is about 3 to 4-fold, so not quite as bad. But if you carry the ApoE4 variant, especially 2 copies, again, you might want to be more vigilant to start a safe, affordable drug that can nip out the amyloid in the bud, nip the deposition of amyloid in the bud. Because all these, in Alzheimer's, it's really the faster you make amyloid in your life, especially over 40 years old, the sooner you get the disease. And it's too late just to treat amyloid after you're diagnosed with the disease because everything's already gone wrong for 20, 30 years since the amyloid deposited. You got to hit it back then. And to do that, you need a drug that's perfectly safe and affordable. I say affordable because if everyone today had the approved blood test that's available to say, do I have amyloid in my brain? And is the amyloid starting these tangles to form and inflammation? 30 to 40 million Americans who don't have Alzheimer's diagnosis yet will find out that they're on their way. Now, they may not live long enough to get it. You know, it takes 1, 2, 3 decades after the first amyloid deposits to get the disease. They may have other genetic factors that maybe quell the downstream effects, so they might not get it. It's not a guarantee you get the disease when you have the biomarker that says you have amyloid. But, you know, if you have high cholesterol and coronary artery disease, you don't roll the dice. You bring your cholesterol down. It would be the same thing here, but the drug has to be affordable and safe enough for CMS to treat 30 to 40 million people. And that's what we're aiming for. That's our, we're seeing the light at the end of the tunnel. We have the drug. It took 25 years to get here. This drug will also be a godsend for the families with early onset familial mutations to guarantee the disease because it will reverse what happens.
So the families, so 30 million Americans, let's just say I've got a magic wand. Everyone gets the blood test. 30 million Americans start taking this pill. And what happens? The people who already have Alzheimer's, obviously you're saying the wildfire, the analogy, the wildfire has started, that there's nothing, this isn't going to help those people, but it's basically going to, does it eradicate Alzheimer's in 30 years from now? Is Alzheimer's cured or prevented? I don't know the right terminology.
Ended, defeated. I don't know the terminology either. We work on it all the time. But yes, that's the vision, is that if this drug works the way we see it does in our lab-based studies and everything from human brain organoids of Alzheimer's, you know, we created a mini human brain organoid that creates in 6 weeks what takes 30 years in a human brain, so we could test the drug quickly there. There are also transgenic mouse models of Alzheimer's. It works in all of them. It does what it's supposed to do in every system where we've tested it. There's no reason. Now, here's why I think it will work in humans. A pharma company will often want the same thing that a biotech company has, but the biotech company, you know, has intellectual property, for example, on the drug. So what pharma companies will do is they might make a version of the drug that's structurally the same class, but just different enough that they get their own intellectual property, right? And we actually want that to happen because every pharma company who makes a drug similar to ours, then we can ask, what do they find? And it tells us what we will find. And they go faster than us. So in this case, Roche made the same type of drug we have, the gamma secretase modulator. It's a similar structural class as our original drug that we started 25 years ago. And we're seeing that their drug is safe. They're achieving their goal of lowering the right forms of the amyloid beta protein in the brain, the toxic forms. They even just announced at a meeting a couple of weeks ago that they treated these patients with familial early onset Alzheimer gene mutations. This is a large family in Colombia that has a presenilin-1 mutation, one of the genes we helped find. And they showed that it reversed in the young carriers what the mutation was doing wrong. So maybe that's like ending a death sentence for them. So we have kind of a preview into the future for our drug by seeing what pharma companies are doing with drugs that are similar to ours, that are derived based on ours, but carry their own independent status in terms of intellectual property. So we hope, we want pharma companies to copy our drug, and so, because we don't care if it's ours or theirs. We're doing this to help to just get it to people. If a pharma company learns from our structures and makes one that works, great. it also paves the way for our drug, which was the original. I like to say Coca-Cola versus Costco Kirkland Cola. So,
Right. And in your career, I mean, we've been in the ALS world for 9 years, 9.5 years here. There's always kind of something that's, oh, this is the next thing coming. This is the next thing coming. And obviously for ALS, it hasn't happened yet. I imagine over your, a long career, you've seen a lot of things that were promising. Has there been anything to this magnitude?
well, we have two approved Alzheimer drugs that are antibodies that help clear the amyloid out after it's already there. So I like to say, picture your brain filling up with amyloid like a kitchen sink, filling it with water. And the drugs that were just approved, it waits till the sink is full of water and maybe there's some damage, and it unclogs the drain. But you do that after the amyloid's already accumulated and it's already, set off this cascade of tangles and inflammation. So you'd like to do it earlier. And the problem is the antibodies are very expensive because you have to have MRIs to make sure your brain isn't swelling or hemorrhaging. So it costs 50 to 100,000 per patient. But the antibodies that are approved do very nicely wipe out the amyloid. Then the amyloid comes back afterwards, slowly. Our drug is different. We turn off the faucet, we turn off the tap. So it's kind of like the way statins work for cholesterol. They turn off the production of the cholesterol. So our type of drug would be one you'd want to use before the sink is overflowing, before you have to unclog the drain. When you know the amyloid is going to begin in your brain based on your genetics or the biomarkers say, yeah, you're right at the very earliest stage. The amyloid's just starting to be made. Let's turn off the tap. And that's what our drug does. So it's a little different. Now, I've also been involved with ALS. I don't know if you know this, but way back when I built the first genetic linkage map of chromosome 21 to study Down syndrome, and it's also how we found the amyloid gene. It's on 21. It explains why Down's patients get Alzheimer's pathology later in life. The ALS world, Tipu Sadiq, Bob Brown were using our chromosome 21 map to look for genetic linkage to ALS. And using our map, they found genetic linkage to chromosome 21. And then we actually worked actively with Bob Brown to find the first familial ALS gene, SOD1. When Bob and his group found a genetic marker, it was called DB1 for Denver, Boston 1, that was tightly linked to ALS, familial ALS. We showed in our lab that it was right on top of the SOD1 gene. And we actually worked with Bob Brown to amplify parts of that gene. And then that led to the mutation, the first mutation in G93 in SOD1 ALS. So we were only middle authors on that paper. I think there were 35 authors, and my postdoc and I who did this were probably authors 17 and 19, and that's okay. But technically, we were the ones who figured out that SOD1 was the gene first with our physical mapping, and then the mutation assessment was done in Bob Brown's lab. So that was nice. The other thing is I'm scientific co-founder of a company that's still around called Amylyx. Amylyx, these were two kids one was I call them kids, they're grown up now, but one had just graduated Brown, the other was a senior, and they came to see me because they were Sigma Chi fraternity members at Brown, and I was a Sigma Chi in college. And I had just been inducted into like the Sigma Chi Hall of Fame, and they read about my work. So they came to see me with this idea of how to prevent cell death with repurposed drugs, and we worked out together a plan. And so I'm like a secret backdoor co-founder because it was a much nicer story to say it was just two kids from college who did it, but I helped them all the way through. I still hold, for disclosure, I hold equity in the company. I'm still the founding scientific advisory board chairperson for them. But that was a nice example that you could repurpose two drugs to create a new drug. And the drug worked fairly well. As you know, in the phase 2 trial, it was approved. When the phase 3 trial in Europe failed, as they promised the FDA, we pulled the drug. But patients were feeling benefit from that drug. And just in terms of what else I'm doing is I'm doing what I call the Amylyx playbook. For the past 10 years, I've been screening every known FDA-approved drug and thousands of natural products in these mini brain organoids that recreate 30 years of Alzheimer's pathology and build up in 6 weeks. And we've identified about 300 different known drugs and natural products that stop this different pathological events over those 30 years that lead to Alzheimer's. And I'm doing trials now. So at the McCants Center for Brain Health, which I direct at Mass General, we have our first trial going using a combination of two natural products that help clear the amyloid away, but also help stop the wildfire, the neuroinflammation. And I have several dozen more of those combinations waiting, and we just did a big fundraiser to start to do as many of those trials as we can. So we're also doing the Amylyx playbook on Alzheimer's at McCants Center.
So is the thought that one of these other treatments is an add-on to what you're doing with turning off the tap water, or is it for trying to help people who already have a full sink?
Okay, so great question. So the real drug, the gamma secretase modulator, is for turning off the tap. The combinations will help unclog the drain, but much more cheaply and safely than the existing monoclonal antibodies. But there's a third thing, which is that they also prevent the damage. They prevent the neuroinflammation that can be caused, which we don't have any drug for yet. So if you think about the kitchen sink, let's say it overflows and then the water goes down to the floor and hits the electrical sockets and causes a fire or damage, right? We're stopping all of it. Basically, if we do our regular drug, that turns off the tap. For the repurposed drug combinations, one will unclog the drain. The other one will make sure that any water overflowing from the sink doesn't do any damage. That's the neuroinflammation.
Got it.
So hit all three.
Exciting.
Yeah.
After outlining the approach when you made that early discovery, what actually happened in the room and what did you feel in that moment?
The early discovery of the Alzheimer genes, the first one was 40 years ago. And there was really no feeling like it. As a scientist, when you make a big discovery and there's about a whole day where you're the only one who knows, it's kind of a pretty magical feeling to know about something that might change the world. And you're the only person in the history of humankind on the planet that knows it for at least a few hours until, you tell your mentor and you tell your other, lab members, hey, guess what, And I've had the, privilege of that happen to me several times with several different genes. But the first gene was the most special. That was the amyloid, the gene that makes the amyloid, which, by the way, when I was a student at Harvard, everyone in my department told me I was crazy for doing that. They said, amyloid's a big garbage dump. There's no gene for that. You're wasting your time. Do something safe. You're going to be a student forever. And I'm very lucky that if you see him back me, my keyboards, I always played in bands. My hearing might be a little bad. So I said, what? Didn't hear you. Oh, and I went ahead and did it anyway.
That's really exciting. When you first helped find genes linked to Alzheimer's, what did you think it would change for families? And when did you realize it would take much longer than you hoped?
Perfect question, Tim. I have to say, when we first found the first three genes, the first one was the protein that's clipped to make the amyloid. The next two genes, and that was in 1986, 87. The next two genes were in 1995, and they were the genes, it turns out, that do the clipping, right? So that's why we knew amyloid had to be important. Here's these mutations that guarantee early onset familial Alzheimer's, and one is the substrate that gets clipped, and the other one are the enzymes that do the clipping to make amyloid. So we were hoping, we'll just figure out how to stop the amyloid, and we'll treat the disease. And then we slowly learned that it was really hard to come up with drugs that could safely stop the amyloid production. Pharma made lots of mistakes with those. And worse, we found out that even when we could clear the amyloid from the brain of patients, they didn't get better. It slowed down the disease, but cognitively, you didn't notice any change in their, memory or cognition. And that's when we started to see more and more brain imaging data over time that showed us, oh, the amyloid starts 20, 30 years before the symptoms. And then that triggers the tangles to form, and they have to build up. And then you only see the nerve cells dying because of inflammation even much later. So it's a 20 to 30 year process. And if you are only hitting amyloid, it's exactly like hitting cholesterol and heart disease. You have to do that 20 or 30 years in advance. You don't wait until a patient's diagnosed here. Because when we diagnose Alzheimer's disease, the brain is equivalent in heart disease to already being coronary artery disease, congestive heart failure, even needing a bypass. We wait that long for diagnosis. That has to change. That's unacceptable. We can't wait until the brain deteriorates to the point of dysfunction to diagnose the disease. The future for defeating this disease will detect you're on your way 20 or 30 years in advance with a blood test or genetics or both. And then you'll take safe, affordable drugs, whether it's the gamma secretase modulator we are now putting into trials, or even safe repurposed known drugs and natural products, the way Amylyx did for ALS, for Alzheimer's. So that's all of our focus right now. But Tim, it was a slow and painful realization to see that hitting amyloid was something that you couldn't just do after a patient was diagnosed. You really had to do that way beforehand to help the patient. It's a great question, and I never was asked that question, but I always wished I was asked that question because it really does explain, when you read about the controversy in the press, oh, these people are working on amyloid, and it's, even just a few weeks ago, someone said amyloid is the wrong target. It's about when you hit it.
As the timeline stretched, was there a moment where you thought we might be looking at this disease the wrong way? What made you question it?
I never thought it was the wrong way in terms of what we had to do. It was the slow realization that it was more important when we do it. That stopping the amyloid was something that you can't wait until a patient's diagnosed. And it's something you want to do decades before symptoms start. Exactly analogous to how I myself managed cholesterol starting back when I was 40 because of my family history to avoid heart disease. And here I am. I'm the first male Tanzi in my immediate family who made it past 45. My dad, his dad, my dad's brother all died by 45 or so with heart attacks. And I have the mutation they had. And what I do, I knew that and started treating my cholesterol with a cardiologist back in my 30s. And here I am in my 60s. So we want to do exactly the same thing for Alzheimer's. Nothing should be a death sentence, just early detection, early intervention, and if necessary, preceded by early prediction by genetics, early prediction by family history and genetics, early detection of the disease process beginning with a blood test, and most importantly, early intervention, nip it in the bud before you get into trouble, before the brain starts to be too damaged. That's our goal. And now I'm thrilled to say we have the drugs that can do this. And the trials are starting. We're seeing the light at the end of the tunnel. It's the most exciting journey of my entire career right now. I should be retiring, but I can't. So. My friends are all retired, all my college friends are retired. Like, what are you doing? Like, this is the most exciting part of my life. Are you kidding? This is why I worked 40 years for this.
Yeah, you can't go now.
Yeah.
You're at the climax.
Yeah.
Is early detection now? What would the cost be if? I don't think I am. I guess I don't know. But if I wanted to go get tested, if I was a high risk person, what does it cost today?
Well, the genetic testing, the most common genetic risk factor is ApoE4. And if you wanted to have a test that does your ApoE4 gene status, plus a blood test to say, are you beginning to have amyloid and the tangles forming in your brain early on, a decade, 2, 3 before symptoms, that test is about $1,500. And insurance doesn't cover it because insurance companies say it's not actionable. In other words, if you have a positive result, we can't tell you, okay, take this drug. We have drugs approved to lower the amyloid, but they're only approved because they're so expensive and they have safety issues. They're only approved for the earliest stage Alzheimer patients. So if you're a person who's cognitively fine, you have a family history, let's say you paid $1,500 out of pocket and you find out if you need that drug now, well, you can't really get that drug. I mean, it's the two drugs that are approved are just for early stage patients. But if we have drugs like the ones I'm developing right now and going into trials now that are safe, affordable, it's a different future. Then insurance companies will say, oh, that test is actionable. Let's pay for the blood test. Let's pay for that $1,500 for the blood test. Or it'll probably be cheaper at that point because of CMS. And let's find out who needs this drug because, man, Alzheimer's is an epidemic that is going to single-handedly wipe out Medicare and Medicaid because we're all growing older and our lifespan has far outpaced our brain health span. So as more and more people over 85 make up our population, well, 30 or 40% of them are going to have Alzheimer's. So we're all living longer thanks to modern medicine, but our lifespan is far outpacing our health span, especially our brain health span. HHS knows this. So as soon as we get something that's safe and affordable that can treat up to 30 or 40 million people who have a blood test and find out they need help decades before symptoms, they're going to help us make that happen.
And I'm certainly not anti-medicine, but I always like to hear if there's natural things, diet type things that people could do. Nothing. Now I'm not saying it's going to be as effective as the drug you have, but if there was somebody who's high risk that's listening to this now and says, I don't have $1,500 for the test, and even if I got the test, they're not going to cover me anyways. Is there things or lifestyle things people could do?
Yeah, I'm known for an acronym that I came up with, and I'm actually finally writing a book on it now with Random House Penguin called SHIELD. And SHIELD, each letter starts with what you need to do in your lifestyle. S is sleep. So when you sleep, the brain actually clears the amyloid out. So let's say you're especially over 40 and the amyloid starting to deposit in your brain. Sleep is like a rinse cycle. It helps get the amyloid out. So, 7 to 8 hours of sleep means you'll have enough deep sleep to dream sleep cycles, and each cycle is like a rinse cycle for amyloid. H is handling stress. And there, basically, stress causes the production of cortisol in the brain and inflammation. It kills nerve cells. So anything you can do to deal with stress, whether it's a meditation practice or just trying to be more mindful in the moment rather than obsessive and worrying about things, anything you can do like that, handling stress. That was the toughest chapter I'm writing in the book right now, is telling people how to handle stress. I is easier. I is interaction, right? Loneliness, being alone and not liking it, having no social interaction increases risk for Alzheimer's 2 to 3-fold. So staying interactive with others, even when you get older. Checking your hearing, because if your hearing goes, you tend to be more isolated socially as well. E is exercise. 20 to 30 minutes of just enough exercise to get your heart rate up 50% every day. When your blood flow is flowing faster, it actually causes amyloid in the brain to be digested. We figured out the exact process of a little hormone that's released by the muscle. When you exercise, it goes into the brain and triggers enzymes to break down the amyloid. It's amazing. And exercise also triggers the birth of new nerve cells exactly in the parts of the brain that are most vulnerable in Alzheimer's disease. So exercise is big. L is learning new things. So every time you learn something new, you make new connections between nerve cells, synapses. And at the end of the day, what causes Alzheimer's is loss of synapses. So the more synapses you make every day by learning new things, the more you can lose before you get into trouble, right? Synaptic reserve, we call it. So right now, your listeners, if they're learning something new, they're being protected from Alzheimer's. If I'm putting them to sleep, I'm still protecting them from Alzheimer's. So either way is good. D is diet, and I would say diet's number one. The bacteria that live in your gut microbiome, are what keep the brain healthy and help control the removal of amyloid. So every day, the more of a plant-based diet you can have. I'm not telling people they have to be vegan. I'm vegetarian myself, but every day, no matter what else you eat, if you can avoid the ultra processed foods, keep the sugar, fat, and salt low, and every day at least have some plant-based snack or meal, that keeps your bacteria in your gut happy, which then help clear the amyloid in the brain and reduce inflammation. So that's SHIELD.
Yeah, that's great. I also too want to say this, and we can, I don't know if this will be in the episode, or we can cut it out if you don't, but I almost wish we had started with having you explain what Alzheimer's is to somebody like me. You two guys are off the chart smart. I'm not. So I almost feel like if we start the episode or having something where maybe you have on your website or something like, what is Alzheimer's? In as in most layman terms possible. Like I know what it looks like when somebody has Alzheimer's, right? And I know like, but at that point, it's pretty far down the road and all that stuff. It's like if you were explaining to my 7-year-old daughter what Alzheimer's is, how would you explain it?
So Alzheimer's disease, in terms of the symptoms, is being unable to remember things, recall things, keep track of things in your life. And it gets so bad that you can't live a normal life anymore. So in other words, as you get older, we might forget names or to forget what something is called. We have the senior moment, so to speak. But Alzheimer's is where you have so much trouble recalling things, with memory, with keeping track of things, that you can't live a normal life anymore. The cause of it, one step back, is that you're losing the connections between your nerve cells in the brain, your neural network. 1 billion nerve cells making trillions of connections. And in Alzheimer's, those connections start breaking down and going away. I think about our neural network like a tapestry that we weave our entire lives. With every experience we have, every memory we make, in your neural network, there are connections being made between nerve cells, tens of trillions of them between a billion of neurons. And Alzheimer's comes and unweaves that tapestry little by little. What causes it to be unwoven, one step back, is inflammation in the brain, just like inflammation in my knee, because I played too much basketball my whole life and had to get a knee replacement. What causes the inflammation? Well, that's the plaques and the tangles, the sticky material that accumulates, especially after 40 years old. And the faster you make the plaques and tangles, the more inflammation you get. But that process from plaques and tangles to inflammation to the breakdown of your neural network takes 20 to 30 years. That gives us a window of opportunity to treat early. That's exactly what we're doing right now with the drugs we're developing, whether it's new drugs or repurposing old, safe FDA-approved drugs and natural products. A lot of the natural products we're finding work great. At the McCann Center for Brain Health, our trials are all on safe, known natural products that we found in our mini human brain organoid model of Alzheimer's, the size of a piece of rice. We found it, which natural products stop the amyloid, the tangles, the inflammation, and those are being tested in patients right now. And I take those natural products myself because I'm not getting any younger, and I share them with people, but only with a disclaimer that says, we have no clinical evidence they work yet. So it's only based on what we've learned in the lab and mini brain organoids, but I take them because they're safe. And so I do provide a list of those if asked to family and friends with a disclaimer that says, we don't have clinical evidence yet. This is not official clinical advice from me or Mass General Hospital. But here's what I personally take based on what I learned in my lab. And we're doing the clinical trials now, so maybe we'll have that evidence soon. So that's basically how we do it.
Yeah, that's helpful. That's really helpful.
When your idea becomes widely accepted, does it get harder to challenge it? When did you notice that happening to you?
Well, there's a quote from Schopenhauer, right, that says all new discoveries, especially the big ones, go through 3 stages. The first stage is they're ridiculed. We've been ridiculed. I was ridiculed as a student, thinking that amyloid, there'd be a gene for amyloid and it's involved in Alzheimer's. The next stage, after being ridiculed, is they're violently opposed. Okay, we've gotten past that, although there's still some violent opposition about whether amyloid causes the disease. Again, because it starts very early and you can't just treat, I went over the controversy. And the third stage, after being ridiculed and violently opposed is it's taken to be self-evident, right? And at that point, they forget all the people who first did it. And the people who take it to be self-evident get the credit, which is fine. So I would say that at this point, if you asked any Alzheimer scientist who's been in the field long enough and really knows the field and reads the papers, they would agree with me. If you ask a journalist who's trying to get clickbait to say all these scientists are stupid and doing the wrong thing, they'll disagree with me and they won't know why. So it's still a challenge. It's still a challenge to convince people that hitting amyloid safely and early in life will stop this disease. But all of the science, from genetics to model systems to brain mapping, postmortem autopsy results, everything says we're right. And we just got to stick with it. And when we get the drugs that actually stop preventing this disease, they'll say, oh, yeah, of course. We always knew that. But for now, we have to fight.
You started growing brain cells in the lab. What were you unable to understand before that made you try something so different?
Man, Tim, you do ask the right questions, I got to say. There was a big controversy about whether amyloid causes the disease, because when we put these genes we first found that cause more amyloid to be made, and we put them into mice, the mice would make the amyloid, but they wouldn't make the other main pathology of disease, the tau tangles. So there were, for a decade or more, there were fights at meetings saying amyloid doesn't cause the disease because in the mice, you don't get the tangles. And then only about 10 or 15 years ago, we learned something very important. Mice cannot make tangles. So when we put in the amyloid in the mice, of course, they didn't make tangles because they weren't able to make them. They don't have the right forms of this protein called tau to make tangles. So my colleague Du-Yan Kim and I said, let's do this with human cells. But we need to make a mini human brain that mimics the brain, grow the human nerve cells with the other brain cells in a gel-like matrix, because the brain is like gel, and put the mutations in there. And now with human nerve cells, not mouse nerve cells, let's see if it works. And then in 2014, we published a paper in the journal Nature showing, yes, in a mini human brain, organoid or assembloid as they're sometimes called, we could see the amyloid plaque causes the tangles. Controversy over. So that's why we invented the mini human brain organoid models, is because you cannot, to this day, replicate Alzheimer's disease in a mouse model. It doesn't work. And that's allowed us to do the drugs.
Has there ever been a result that looked too good where you thought this cannot be the whole story?
In genetics, sometimes, when we're looking for new Alzheimer genes, sometimes you'll find a new gene that looks like it describes, it accounts for most of the disease, and it's just a type 1 error. So in genetics, you get a lot of that. So I've learned to never be too excited by new genetic findings, because 50% of the time they go away. They don't last. I guess other than that, it's been such slow, incremental success in Alzheimer's disease that other than genetics, we really don't experience that a lot. Now, maybe I can be accused of that 2 years from now, when I think that the drugs that safely lower the amyloid.
At some point, you began looking beyond the brain itself. What made you think the cause might not be where everyone was looking?
Well, if we ask, how does amyloid accumulate faster in some than others to get you to Alzheimer's faster? You can look at genetics. You can look at lifestyle, like the aspects of SHIELD that I explained. But you can also look at your exposure in your environment. We now know that microplastics that's in our food supply can drive Alzheimer's pathology faster. Infection, this was out of the box. We found out that infection can drive Alzheimer's pathology faster. In fact, one of the fringe hypotheses for which we are still ridiculed is that we believe Alzheimer's pathology evolved as a host defense system for the brain, because amyloid plaques form to trap microbes. tangles we just showed in a new paper in Nature Neuroscience. The tangles prevent herpes virus and other virus from spreading in the brain. Inflammation kills the microbes. So we're looking a lot at the exposome. What are you exposed to every day in your life that combined with your genetics and your lifestyle might be driving amyloid either more slowly or more rapidly in your brain in terms of determining how fast you get to Alzheimer's disease itself.
You have said that something causing damage might also be protecting us. When you first said that, how did people respond?
The same way they respond today, they roll their eyes. I mean, that hypothesis is clearly still in the ridicule stage. But, you know, by the time it gets to the taking to be self-evident stage, I'll probably be dead. That's fine. But the evidence is there. Amyloid beta protein that makes up amyloid is a classic antimicrobial peptide. We showed that. We showed that the tau protein that's induced by the amyloid prevents tangles from spreading from one nerve cell to another. We just showed that very clearly. And inflammation, well, the role of inflammation is to fight microbes. And at the end of the day, you know, the plaques and tangles get you to inflammation, which is the ultimate antimicrobial response of a tissue and of the brain. So I think we're right. You know, it will say to people, be careful with recurring infections. If you get cold sores all the time, get on an antiviral. You know, there are safe antivirals like acyclovir and valacyclovir that prophylactically prevent herpes cold sores. But if you're getting more than 4 a year, well, we showed that herpes virus drives plaques. We showed that herpes virus drives tangles. So we also showed that in Alzheimer brain, postmortem brain, you see more periodontal bacteria from gum disease than in control brains of people who are older but didn't have Alzheimer's. So, you know, floss your teeth, you know. I got a whole pack of these right on my desk. Floss your teeth whenever you can. Because the periodontal bacteria, when they get into the brain through the cribriform plate, can drive amyloid deposition in tangles and inflammation. So, you know, when I tell my colleagues this, colleagues I really respect and have known my whole life, good friends of mine, they kind of go, oh, yeah, okay, yeah. But they don't really believe it. It's okay. I think it's more important to think out of the box and look for acceptance.
What is something people think they understand about Alzheimer's or about your work that is actually wrong?
Yeah. You know, it's important to, you know, to always think we don't know everything, right? So, I mean, you know, I think a lot of the ideas we came up with first, people originally thought were wrong. And you guys got to stick with it, you know. Going back 40 years, we're thinking there'd be a gene for amyloid, that the entire neurobiology department at Harvard Medical School said no. Now it's obvious. You know, saying amyloid causes tangles, even though it didn't happen in mice, and then finally showing it in human organoids. People said no, now they have to say yes. Almost everything I've done in my career, people doubted and said no in the beginning. And you got to follow your intuition. You got to follow what you really think is going on, you know, with a good dose of evidence. But let's face it, if all science was only based on what people think and purely logic and rational thought, we'd be nowhere. You got to take a chance and you got to follow your intuition as well. And you can't be afraid to be criticized, right? The need for external validation is the worst enemy of a scientist.
When the data does not give you a clear answer, how do you decide what to believe or pursue next?
Well, you know, it's interesting you ask that because I remind my students all the time, your job as a scientist is to not prove what you think, but to disprove it. So I tell people, find that balance where you let your mind just go ad absurdum off, like the wildest ideas you can come up with. And then once you find one you really love, right? Remember that you date your hypothesis, you don't marry it. And you're always ready to break up with it. You go into the lab and your goal is to show this hypothesis, idea you came up with that you're so passionate about. Your job in the lab is to show it's wrong. You design all of your experiments to disprove yourself. And that's how you get the right balance of new innovative ideas, but keep yourself honest not to delude yourself. And I know scientists who have great out there ideas, out of the box ideas, but they spend their whole life trying to prove it. And it probably could have been disproven in a week. So the goal of a scientist is to show you're wrong, to prove the null hypothesis. And what usually happens is it's not black and white. You come up with an idea, you do the experiments to disprove it, and some of it's right and some of it's wrong. And you take the little threads of truth of what's right and learn from what's wrong, and then plan the next set of experiments the same way. So the hypothesis keeps evolving. But each step along the way, your aim is to show that your next hypothesis is wrong and disprove it. And then you're not wasting your time. Worst thing you want to do as a scientist is waste your time for 40 years on a hypothesis you could have known was wrong in a year.
You have had major breakthroughs. Did your success in Alzheimer's ever start to feel like pressure instead of relief?
It's always discovery in science and research, and the whole process is pure joy, right? I tell scientists that if they want to succeed in science, you need PDJ. P is perseverance, stick with it. D is devotion, really be devoted to what you're doing. And as important as J, joy. You have to really enjoy what you're doing. Where there is not as much joy is, and where the pressure is funding. Funding is always the bottleneck, especially right now. Funding is really tough. So, the main pressure on us and the most discomfort comes from trying to keep your lab going, especially during tough times and trying to get funding. And these are tough times right now, but we get through it. I tell my lab members, when the going gets tough, the tough get going. And my other thing I tell them is, when you're going through hell, keep going. And that's what we're doing. So, and there's always ways to raise money. If one source of money dries up, which right now federal funding is very low, then we move on to philanthropy. We move on to donors. We move on to other federal agencies, even beyond the NIH, that can help us, something we're doing right now as well.
You study how the brain responds to damage and disease. When you look across Alzheimer's and ALS, what do they seem to have in common that most people would not expect?
The end game. So in other words, whether it's Alzheimer's disease, Parkinson's disease, ALS, Huntington's disease, CTE, there are different ways to first get misfolded proteins of different types that gum up nerve cells and start to kill them. But at the end of the day, it's that process that triggers neuroinflammation that causes 10 to 100 times more nerve cell death than what triggered it. So like in Alzheimer's, plaques and tangles, if they cause 1x cell death, the neuroinflammation triggered by it causes 10 to 100x. And that's when you get the disease, the wildfire. So I'd say in all these diseases, it's the wildfire at the end, it's similar, that leads to enough nerve cell loss in some particular brain region where those misfolded proteins lived to finally get a disease. So, and depending on where the original nerve cells are, where you get the misfolded proteins that trigger the disease, that might be the spinal cord, it might be the motor neurons, it might in ALS, it might be the locus coeruleus and dopaminergic neurons in Parkinson's disease. So depending on where it happens, that's the brain function that's going to be affected, movement or cognition, et cetera. But what we see similarly across these diseases is you begin with a misfolded protein. Like in ALS, it could be SOD1, it could be TDP43, it could be FUS. In frontal lobe dementia, it's the tau tangles first. In Alzheimer's, it's the amyloid first causing tau tangles. In CTE, it's banks to the head causing tangles, and so on. But at the end of the day, these processes trigger eventually enough inflammation in the brain to cause exponential 10, 100 times more cell death, and that leads to symptoms. So if we come up with therapies and drugs and medicines that stop neuroinflammation, those are the ones that we hope could be used across the board, even if it was found in Alzheimer's, to then try them in Parkinson's, ALS, Huntington's, et cetera. And that's something we're doing at Mass General right now. And I'm screening natural products and repurposed drugs to stop neuroinflammation. I share those with Merit Sokovich, who's the head of our Neuroscience Institute, who's the one of the leading lights in ALS, because she wants to try them in her ALS trials as well. So now, if I have a drug that just stops amyloid, that's not going to help in ALS. That hits the match. It's a different match in ALS. But if I have a drug that stops the wildfire, I'm going to try it in ALS, Alzheimer's, Huntington's, and then we use those across the board in trials. And that's something we're doing right now between the McCann Center, which I direct for trials, and the Healy Center that Merit Sokovich directs for ALS, all at Mass General.
And so it sounds like something, I mean, that, I guess, I don't know. Early on, they all sound similar, right? You've got the folded protein. It's matters about where it's starting and what's causing it.
What
the way you just described it, I did not know that. I didn't know the other diseases also. I knew ALS was the folded protein and SOD1 and TDP43, but I didn't know the other things had it. It sounds like they're actually more linked than I realized, I guess.
Yeah, so you have different proteins that can have mutations that make them misfold and form some sticky material, and that causes cell death in different ways. Amyloid starts in Alzheimer's. Alpha-synuclein causes the Lewy bodies that start in Parkinson's disease. And ALS, it could be SOD1 or TDP43 or other genes. In frontal lobe dementia, it's the tau protein, but it can start with other proteins. So and by the way, if I really want to be ridiculed, I'll tell you the following. I believe in all these diseases, the reason why these misfolded proteins and the mutations that cause them remain is they all played some role in brain host defense. We found that TDP43 has antimicrobial activities. Another ALS and frontal lobe gene, FUS, has antimicrobial activities. My daughter goes to Milton Academy and just graduating, and they had this senior poster session the other night. And one of the students who was in my lab for an internship, she's a senior in high school, and she's going to MIT next year. She had a poster. I couldn't believe it, showing that FUS, the FUS protein, based on what she learned in my lab about the antimicrobial aspects of these disease proteins, she showed that FUS had antimicrobial aspects. I said, you have to come back to my lab and do this. You publish a paper. We showed that TDP43, alpha-synuclein, and mad cow or CTE, prion proteins, all these misfolded proteins, when they're misfolded, can help fight microbes. So, talk about eye rolls, but I think that in most neurodegenerative diseases, where the misfolded protein is the beginning and the neuroinflammation at the end takes you out, that the reason why we have these misfolded proteins is they were conserved because they played a host defense role in the brain tens of thousands of years ago, maybe, in the beginning of, going back even before hominins, going back to early non-human primates as well. Now, whoever's listening in the field, when I say that, that'll be a new level of ridicule, but it feels right.
As you move toward treatment, what slows down progress? People with these life-changing diseases need help now, but progress is slow. What is slowing you down today? And what does that gap feel like when you think about the families waiting?
Oh, one word: money, funds. The bottleneck right now, the only constraint on us is how fast we can do things based on how much funding we get. And it's never been worse than right now. Like in Alzheimer's disease, the NIH actually has money for Alzheimer's, but they're only funding, instead of in the old days, they would fund 15% of grants, which is pretty good. And they give 1 year at a time of funding. Well, last year, they only funded 3% of grants, but they gave all the money out at once because that requires less management of the money year to year. So only 3% of Alzheimer grants got funded last year, even though the money was pretty similar. And I think it's just because of trying to, make it more streamlined with the need for less people to do it. And so we hope they'll go back with the same amount of money to fund 15% of grants rather than 3%. But frankly, Alzheimer labs are going away. We're seeing labs dry up. We can't get people from other countries to come work here anymore. People who are here from other countries, many of our scientists are going back. There's a brain drain going on. There are huge issues right now until we get, things going back to where they were. And so it's money, it's budget that's constraining. That's the number one obstacle.
Your work gives people hope. Have you ever worried that hope might come too early?
Yeah. I think part of our job is to not give people false hope. And so, when money is low, it causes some scientists to make exaggerated press releases about how important their findings are that might give false hope to patients. And it's unfortunate because, in order to raise money, sometimes people will exaggerate, and they'll do this more when there's not enough money to go around, like right now. So one of the things we teach in our labs, in the unit I direct and the center I direct, is to never overstate what your data mean, to never inadvertently give families false hope about the implications of your new discoveries that you're publishing, where there might be a press release involved.
It's a really hard line on the other side of the table, right? It's like as a family going through not Alzheimer's but ALS and a similar enough thing. It's like you want to hear about the stuff, even if it ends up being a miss. Like you want to hear about the good stuff going on, because there are times where it feels like nothing's happening. And that's not the truth, right? Obviously, there's people like you and people like Merritt and people like, all kinds of amazing doctors and scientists and researchers out there that are, attacking all these diseases. But yeah, it's a fine line, because you don't want to be, a salesman, right? And saying like, oh, this is the one, this is the one, this is the one. But it's good for people to hear it. And I think part of it is like distribution, like when you release a paper, like a finding in your study, it's not, I'm not saying you specifically, I'm saying all medical. It's so dense for people who don't know the terminology or don't know what it's like to read a medical paper. So yeah, I don't know. It's hard. I like hearing the stuff, even if it ends up not working, because it's good to know that we're swinging,
Yeah, no, you do. You have to find the right balance of letting people, like you said it perfectly, Troy, that you need to let people know that progress is being made, new discoveries are being made. But at the same time, you can't make them think there's going to be a cure next year because you found out how some gene mutation leads to a change in a cell. It's a slow incremental process. I mean, think about it. My own career, every experiment I've done since the early 80s has gotten me to where I am today. I mean, I remember the first time, after I worked with Jim Gasella, where we found the Huntington's disease gene. It was the first time genetics found a disease gene. I was the hands. Jim was the brain, Jim Gasella. I nominate him every year for the Nobel Prize because it was the first time a disease gene was found with genetics. But I remember it was 1982, I did my first experiment to show, in these early onset familial Alzheimer families, onset under 60, I saw linkage to some of the same markers on my chromosome 21 map where we saw ALS linkage that led to SOD1. And then that led to the amyloid gene. And then that led to these other genes that help make amyloid. And then we realized that amyloid, is something you have to hit early, not when you're already diagnosed. And I said, well, what do you hit when you're diagnosed? Oh, you have to hit inflammation. And then in 2008, I found the first gene in Alzheimer's that causes neuroinflammation called CD33. But CD33, which I found in 2008, think about how long it took to get to where I am for amyloid, where we found that gene in 1986, right? So there's a long gap between finding the gene that tells you the target to actually translating that finding to a medicine that will work. That's why we jumped from mice testing one drug at a time for a year to these mini brain organoid models that actually better represent the disease, human brain organoids, where drug discovery is 100 times faster, 100 times cheaper, because you can do 100 drugs at a time in 6 weeks rather than one at a time in a year. And that's what we've been doing in Alzheimer's disease. And luckily, because we did that, and we learned from Amylyx and their ALS drug that you can repurpose drugs to make new drugs, I'm hoping things will go much faster now. And so the genes we're discovering, and believe it or not, there are now 170 Alzheimer genes, 170. And all these genes, now we're using AI. I just used up all my credits on Perplexity. And you get so many credits per month. And I got
totally
Perplexity
get it.
Pro, but it said, you are out. I was just going to get my answer and said, you're out of credits. And I had to pay for more damn credits. But where I can put in like 170 Alzheimer genes at once and say, come up with the pathways, divide these into pathways. Now divide these into molecular mechanisms. Now suggest for me which drugs that are already approved or natural products might work. And sure enough, it came up with ones where when we did our screening ourselves, came up, right? So AI is going to make a huge, look, there's a lot of crap with AI too. Crap in, crap out, no doubt. But AI is going to make a huge difference here as well. So I think this whole process is going to start to go exponentially faster. And by the way, the drugs and natural products that the AI just spit out, because I did pay for more credits, in addition to the ones we already found by just screening, the ones we didn't find by screening, we're testing those now. So this is, it's a whole new world.
Yeah, that's interesting. And how long would that have taken you to do before?
Years.
When someone challenges your work publicly, what is your immediate reaction before you have time to think?
Yeah, I don't care. When if you're trying to do things 5 or 10 years in advance of others and you're trying to be innovative and think out of the box, you got to develop a thick skin. And the only time I care about whether people believe me is whether it's going to affect my ability to publish papers, which affects my ability to get funding. Do I really care what people think about me or about my ideas and hypotheses? Do I need them to think I'm a good boy and a good scientist? No, I couldn't care less, right? I just got to have enough acceptance to get papers published and get my funding. But when someone, criticizes, it just happened a couple of weeks ago, someone criticized something that we published. And I just say, go back and read the paper again. That's what I tell them. I go, we publish careful papers. Go back and read it again. Let me know what you think. Then we can talk about it. Because, if you publish a good paper with sound results and you do it the right way, test your hypothesis to disprove it, you publish it. If someone didn't read it closely enough and they just want to go through their own bias to say what they think, that's their problem, not mine. I just say, go read the paper again and let me know. Email me. Tell me what you don't like about it.
After spending your life studying the brain, has it changed how you think about your own decisions or relationships?
Oh, yeah. I would say, I practice my SHIELD protocol for lifestyle religiously. I get 7 to 8 hours of sleep. I try to handle my stress, actively. I stay socially interactive. I exercise every day. As a scientist, I'm always learning new things. And for diet, I'm already been a vegetarian since college, but, I try to really emphasize the plant-based diet. Because we did a trial with Dean Ornish, famous doctor who came up with the cardio healthy diet. And we did a small trial that was mainly plant-based diet for 20 weeks, 25 Alzheimer patients on, 25 not. And the results were astounding. It's too small a trial to go crazy, but with a plant-based diet after 20 weeks, the 25 patients who are on it, by some cognitive tests, they even got better. Because when your gut microbiome of bacteria love plant-based foods, it's what makes them healthy. And when your bacteria are healthy, they make metabolites that go to your brain and both clear amyloid, blow out the match, and quell the wildfire of neuroinflammation. So we're doing a bigger trial on that right now. And in fact, the McCann Center just joined with Erickson Living to start a pragmatic trial in senior living centers where they'll be getting aspects of SHIELD and diet, et cetera, to try to to try to improve their brain health and try to slow down disease as well. So I practice it myself for sure.
Is it just the red meats and stuff like that? Like, is chicken? You always hear like chicken's one of the healthiest things, and veggies in the
I'm not saying
Mediterranean
that. I'm not
diet.
saying that. Yeah, I'm not saying it's the meat and chicken and fish that's necessarily hurting you. It's the lack of plant-based diet that hurts you. So no matter what you eat, I don't care what you eat. If you're eating your steak, your chicken, your fish, whatever. Three rules: ultra-processed foods, cold cuts and really processed stuff, stay away from it. Number two, be careful with the sugar, fat, and salt. We evolved to love them because they helped us to survive when we only lived to 25 years old. Three is no matter what you eat during the day, take a break to have your medicinal food. Medicinal food is plant-based products, fruits, vegetables, nuts, whole grains, seeds, right? Think of them like medicine. Take them like medicine. Whatever else you eat, I don't care. Just as, as long as it's not too processed, not too much sugar, fat, salt. But take every single day, just keep track that I have my medicine, that I have my plant-based diet foods, because they actually actively help you. I care more in your diet about what actively helps you more than what actively hurts you. And the only thing that actively hurts you is really ultra-processed foods and excess salt, sugar, and fat. Because what's good for the heart is good for the brain. And now we know equally true is what's good for the gut and the bacteria that live in your gut. 8,000 strains, trillions of bacteria. You rely on those to keep the rest of your body healthy, especially your brain through the gut-brain axis. And that's never been more true. And we published many papers on this. In fact, Cure Alzheimer's Fund, the foundation I've been working with for 20 years that gives out $30 million of research for Alzheimer's every year. And the cool thing about them is every dollar they take in is a dollar out, no overhead. They're board members and pay the overhead. And they're funding a whole consortium just on the gut microbiome and how it keeps the brain healthy, including some of the top gut microbiome scientists in the world. And when I look at this trial we did with Dean Ornish, the lifestyle trial where Dean delivered plant-based vegan meals and snacks to the patients at home, so they and the spouse could have it. When I see the results of that study, and Rob Knight, the famous gut microbiome scientist, looked at the gut microbiome changes, it's amazing what we're learning right now about how a healthy gut microbiome of bacteria can keep your brain healthy. And it means mainly just taking time every day to have a plant-based meal or snack. Doesn't matter what else you eat, as long as you stay away from the junk food.
Shifting to something more personal, I have found a lot of stability in my life through faith, and I'm always curious about how others approach it. Has your work ever pushed you to think differently about what you believe beyond science? Religion has given me a North Star that I can always work towards.
Yeah. As a scientist, I believe that to not believe in God is illogical. Because as a scientist, am I supposed to think everything came out of nothing? And where everything came from, I call that God. I call God the overall existence and awareness of the universe that I'm part of. And so when people say, do you believe in God? Yeah. And they say, well, how can you believe in God? You're a scientist. I'm like, well, I don't believe in some guy with a white beard up in the sky. I believe that, God is what we're all part of in awareness and existence, because otherwise, look around. Where did this come from? You're going to tell me, oh, yeah, it just came from nothing. You're a scientist and all this came from nothing? What kind of scientist are you? So that's how I believe. and I pray to God because I believe that there's a intention, a level of intention and momentum that comes from all that is God. I think that North Star, the belief in something bigger than we are that we're part of, and I call that God, is always important. And our humility in that regard is important. And I remember when I was working with Jim Gasella in the early 80s, and Gasella, he was 25 and I was 20. And this is 1980. And we wanted to find the gene for Huntington's disease, and a movement disorder, neurodegenerative movement disorder. And no one knew what caused Huntington's. And so we were testing the first we pulled out the first genetic markers in the genome ever. Just 12 of them. 12 random pieces of hay from 23 haystacks trying to find a needle. And I was the hands. So when we were doing this, I was praying. I was saying, I was literally closing my eyes and saying, God, let me pull out the right one. Because I was like literally pulling out these little plaques from a dish, what are called viral plaques, where the little human clones were. And I was like saying, guide my hand. And it was miraculous. We found the gene. With the first 12 pieces of hay, we found the needle. Maybe there's something there. You got to experience things you can't explain logically like this to really know and believe in something bigger than us that can help guide us, like God. And I do believe that.
Before we close, I always ask our guests to brag about their family for a minute. Rudy, please go ahead.
Well, my wife, Dora, just retired, but she has been an amazing cell biologist studying Alzheimer's, and the drugs she first developed called ACAT1 inhibitors, I'm still developing now in my lab. A lot of her people from her lab joined my lab and she retired. And she's I always say she's a much better scientist than me, much more meticulous, much more detail oriented. My head's in the sky sometimes. And then our daughter, Lila, is just graduating. Milton Academy. We live in here for that school. And I always thought she was going to apply to Harvard, early decision. And she spent 1 month at Northwestern, and she fell in love with it. And so now she just got accepted in December to early decision for Northwestern, where she's going to go and study journalism and pursue also her drama and theater activities. In fact, tonight she's in the first night of her last play ever at the high school at Milton. So we're going to that performance tonight. But she's an amazing actor, and now she's doing for a senior internship with the Boston Globe with one of the journalists there. So very proud of her too. Thank you for letting me say that.
Now on to our final word segment, where we ask our guests a few rapid fire questions. What was the happiest moment in your life?
When our daughter was born.
What is the biggest adversity you've faced?
Right now, losing lab members because of funding.
What are you most excited about?
About our drugs getting approved and ending Alzheimer's once and for all.
What advice would you give your younger self?
Don't care what other people think.
Do you have any book recommendations for us or for the listeners?
Well, if you're interested in the history of Alzheimer's disease and how we found the first genes and understand the disease even to this day, the first book I wrote was Decoding Darkness, the search for the genetic causes of Alzheimer's, and it really tells the full story from Alzheimer himself up until now in terms of what we learned from those genes and how we found them.
The name of our podcast is Nothing Left Unsaid. Do you have anything you want to say?
Yes, thank you. And I so admire what you guys are doing and the incredible ability to have an interview with Tim. And you guys, I'll just say, you guys asked some of the best, most insightful questions, one after another, that I've ever had in an interview, and I'm grateful for that. So thank you.
I'll send you the $1 for saying that. I'm glad you read that.
No,
part.
I mean it. I mean it. I mean it.
Dr. Tanzi, before we go, at the end of every podcast, we always ask our guests who are people they recommend that we have on the podcast to tell their story and who would be interesting and people you know personally. And actually, this is a funny one because our, I think, third ever episode, maybe a second episode, was Mara Sokovich, and she recommended you. And now we're, 100 episodes later, and we got you. So, or 100 plus episodes later. So, who are a couple of people you know that you think we should have on?
I spent 9 seasons as the brain health consultant for the Patriots, and I don't know how you guys feel about the Patriots, but but I know. But I spent 9 seasons. I actually have 2 Super Bowl rings from that. Would have been 3 except for that last fumble against Philadelphia, where I was like 10 feet away on the sidelines in that Super Bowl. I would say, Bill Belichick has been a big influence on me in terms of his ability to not care what people think about him. I would recommend Bill if you were open to that.
Yeah, no, he'd be amazing.
Rudy, thank you for a lifetime of work that has shaped how we understand the brain and for taking time for walking us through it so clearly today. God bless you and your family. Now, I don't want to take up any more of your time. I want you to get that cure and then join Merit to help fight ALS.
Yes, well, I'm ready to join right now. A little late. My the clinical trial group was doing our new clinical trial on Alzheimer's, and so that's what I'm going to jump to right now. And I appreciate your having me, and this was really fun. Great interview. Really appreciate you guys.
Yeah, thanks so much, Dr. Tanzi. This was awesome.